Rivers and streams are critical to life across the planet. With the ongoing threat of habitat loss and degradation, protecting freshwater is an urgent concern. In 2017, World Wildlife Fund Canada's watershed reports showed that data gaps persist in 66% of Canada's watersheds. Project STREAM works to fill these data gaps and monitor the impacts of stressors on freshwater systems. STREAM is a community-based, Canada-wide biomonitoring project led by the Hajibabaei Lab at the University of Guelph's Centre for Biodiversity Genomics. Through STREAM, we assess DNA sequences from benthic macroinvertebrates, which are small bugs that are sensitive to environmental change and inform us about aquatic ecosystem health. We use a tool called environmental DNA metabarcoding to produce tens of thousands of usable DNA sequences from KickNet samples collected by community groups. We match these sequences to a corresponding reference sequence stored in international databases. Let's follow the journey of a stream sample as it transforms into watershed data. We start with a preserved benthic sample and blend the contents to make a mixed slurry. Once homogenized, we condense the slurry through centrifugation. This prepares the DNA for extraction by pouring off and evaporating the preservative. We transfer a small portion into extraction bead tubes, where it's combined with lysis reagent to break apart the cells and release the DNA. That's when DNA within the lysate can be isolated through an extraction process that removes inhibitors for downstream processes. Next, we amplify the DNA sample through polymerase chain reaction, or PCR. PCR uses the enzyme polymerase to make millions of copies of selected regions of the genome. They're targeted using short synthetic nucleotide sequences known as primers. We target regions of the genome that are specific to each taxonomic group and can be used as a DNA barcode to identify the organisms in the sample. Before we sequence the DNA, we confirm successful amplification using gel electrophoresis. We repeat the PCR step, but this time we tag the primers with special nucleotide sequences that help Illumina's MySeq system recognize the sample. Purification further concentrates the DNA and removes any excess primers or reagents. Now the sample is quantified and normalized so that all samples are equally represented during sequencing. We complete one more step to anneal unique sequence tags that differentiate samples. This allows us to pool all samples and sequence them together. Then we can separate the resulting sequences for each sample by using their unique tags. After pooling, the sample undergoes a final quality control and we proceed to sequencing. As we load the library into our in-house Illumina MySeq, we wait for the magic, or science, to happen. Our lab team runs the sequence files through our analytical pipeline called MetaWorks before sharing preliminary data reports with participating community groups. All the data from those tiny benthic macroinvertebrates are then released onto STREAM's public data portal, where you can find over 1,000 biomonitoring sampling results from coast to coast. STREAM would not be possible without the generous support of our funders, like Illumina, a large biotechnology company and industry leader in the development and application of DNA sequencing. With Illumina's philanthropic support, STREAM continues to expand biomonitoring efforts to more communities and to produce more data supporting freshwater health. Every STREAM sample's journey contributes to our understanding of watershed health and the steps we can take to protect freshwater across Canada.